I've been at CABBI since the beginning, so we're approaching the end of our first 5-year phase. The focus of my research is on the genetics and breeding of Miscanthus, and also of Miscanthus as a genetic resource to improve sugarcane. Miscanthus is a relatively new bioenergy crop, and one of the limitations that we have is that there's really only 3 cultivars that are available to growers, and they're very similar to each other. The U.S. has a lot of growing environments, and so for the Midwest, we would like greater cold tolerance, winter hardiness. For the South, we'd like them to flower later and have more biomass. One of the unique features about CABBI is to produce high oil producing energy cane or Miscanthus or sorghum. And that's a really unique concept because the plants don't normally produce a lot of oil. And yet there has been quite good success in increasing the amount of oil that these plants can accumulate. It's not a final product just yet. There's a lot of work still to be done, but I think there's a very strong proof of concept that is quite encouraging. The sugarcane and the Miscanthus are perennial crops. They're long-living plants, but it also means then the cycle of breeding is very long typically. The idea is using these technologies to decrease the breeding cycle, and that'll be a breakthrough that'll have advantages for many years to come for all sorts of people, not just within CABBI. You know, if you're into plant breeding, you have to be excited about genetic diversity. And here's essentially a wild plant with a few selections that has a geographic range from eastern Russia into the tropics of New Guinea. It's just so diverse, and there's so much interesting stuff going on in these plants, and we've only just touched the surface. This is our main crossing house, and it's very tall sidewalls so we can flower these sugar canes. And really what we're trying to do is use Miscanthus as a genetic resource to improve energy cane. So Miscanthus has exceptional winter hardiness and cold tolerance and low temperature photosynthesis, very exceptional for a warm season grass. And we'd like to move those traits into these canes, which are tropical. And the other thing that Miscanthus has is a lot of disease resistance. So that's our main interest with the canes and the Miscanthus. And then we're also breeding Miscanthus in its own right for adaptation to the Midwest and to southern US. We were looking at the growth chamber experiment where we had a mini core collection of Miscanthus sacriflorus. And so these came from different latitudes all throughout its native range, from 50 degrees, nearly 50 degrees latitude in eastern Russia to around 35 degrees latitude in China along the Yangtze River. And we're looking how long days like 16.5-hour days or short days like 12.5-hour affect how long it takes for these different plants to get to reproductive stage, when they'll go dormant. And then we can use that information to help us time the crosses between the Miscanthus and the canes. So the more tropical and subtropical Miscanthus tend to have higher biomass potential, but they're not winter hardy here. Well, we'd like the best of both worlds, but they won't flower at the same time unless we can manipulate the flowering time, can trick them into flowering at the same time, and then we can put them together. So that's what we're learning to do. Earlier, our harvest with a large machine out in the field, and then sometimes when we have smaller plots, we will cut individual plants and then weighing those dried samples to get the dry biomass weight so we can learn which ones yield more, yield less, so that we can make selections for further crosses, and also to understand the genetics and trying to develop genomic selection models to predict at the seedling stage which one of those individuals would be among the best for biomass yield so we can turn generations very quickly. We have probably the largest germplasm collection of Miscanthus anywhere outside of Asia. We have found that there are 7 genetic groups of Miscanthus sinensis and 6 genetic groups of Miscanthus sacriflorus. And so what we've been doing then is we've been taking these sets of plants that belong to a genetic group, which usually belongs to a geographic region, and we intermate those plants. And then we take those populations, half subfamilies, and we grow them out in replicated trials in the field. And so population size matters, but you know, if it requires a bunch of labor, then there's only so many you can grow. But if it's going to be easy, then you can grow bigger. So you can really make more gains over time. I hope that the research that we're doing here will eventually translate to crops grown in farmers' fields that contribute to a sustainable bioeconomy. It won't be the only solution for energy and products in the future, right? But I think there's an opportunity to bolster U.S. agriculture and make us more sustainable at the same time. And we're in the foundations of that right here.